It's a pleasure today to be here. Thank you to Emmett and thank you to the organisers for this opportunity to tell you about OPTHEA, without an H. Today I'm going to talk about our Phase 2b clinical trial results of OPT302, which is a novel first-in-class inhibitor of VEGF-C and VEGF-D, which we are developing as a combination treatment for neovascular AMD. We are a public company on the ASX and therefore I have a statement regarding forward-looking statements. Okay, so the currently approved therapies for the treatment of neovascular AMD share a common mechanism of action, as we know, by targeting VEGF-A and preventing the interaction of VEGF-A with VEGF receptor 2. These, of course, include ranibizumab, bevacizumab, as well as aflibercept, which is a trap molecule that blocks not only VEGF-A but also the VEGF receptor 1, specific ligands VEGF-B, as well as placental growth factor. OPT302 is a first-in-class novel trap inhibitor of VEGF-C and VEGF-D, which completely blocks the binding of VEGF-C and D to VEGF receptor 2, as well as to the independent pathway, VEGF receptor 3. And used in combination with a selective inhibitor of VEGF-A, we can mediate a more complete blockade of the VEGF receptor pathway. And we can block VEGF-C and D mediated upregulation, which can occur in the presence of a VEGF-A inhibitor. And this upregulation can contribute to suboptimal clinical responses with VEGF-A inhibitors. So today I'm going to talk about OPTHEA's Phase 2b clinical trial. This is a randomised, double-masked, sham-controlled study of intravitreal OPT302 in combination with ranibizumab in participants with neovascular AMD. This was a multi-centre study conducted at over 113 sites across 10 countries in the US, Europe, as well as Israel. The study recruited treatment-naive patients with neovascular AMD according to standard eligibility criteria for these studies. We recruited patients with predominantly classic, minimally classic and occult lesions with a baseline visual acuity of between 25 and 60 letters. We randomised 366 patients into the clinical trial to 3 treatment groups, a control group consisting of sham plus ranibizumab, a low-dose OPT302 dosing group in combination with ranibizumab, and OPT302 at a high dose of 2 milligrams in combination with ranibizumab. All agents were administered via sequential intravitreal injection. They were administered on an every 4-week dosing cycle for 6 months, and we had very good follow-up in the study. The study's primary outcome was the mean change from baseline in best-corrected visual acuity at week 24, with key secondary outcomes looking at visual acuity, as well as OCT anatomical changes, as well as exploratory outcomes and pre-specified subgroup analyses, which I'll take you through today as well. The baseline characteristics and study demographics between patients enrolled into the clinical trial were well balanced between the treatment groups. We had a mean visual acuity at entry of approximately 50 letters, balanced across the treatment groups, with balance in the predominantly classic, minimally classic and occult patient types across all of the treatment groups. 18% of our patients had polypoidal lesions and 14% of our patients had RAP lesions, with a slight increase in incidence of RAPs in the low-dose OPT302 group. The study hit the primary endpoint, demonstrating superiority in visual acuity over ranibizumab in the OPT302 2 milligram dose group. Between baseline and week 24, we saw a mean gain in visual acuity of 14.22 letters in the combination treatment group, compared to 10.84 letters in the ranibizumab control group, which was one of the best performing Lucentis or ranibizumab control groups that we've seen historically. This resulted in a superior vision gain of 3.4 letters in the combination group compared to the control, and it hit a p-value of 0.0107. The low-dose group did not show any significant difference to the control group of 10.8. This is the BCVA over time. We saw added visual acuity benefit of OPT302, evident from 8 weeks, which persisted over the course of the clinical trial of 6 months. We saw a higher proportion of patients that gained over 15, 10 and 5 letters of vision throughout the study treatment following OPT302 combination treatment. We saw further reductions in CST following OPT302 treatment compared to the sham ranibizumab group. And we also saw an increased rate of drying of subretinal fluid and intraretinal fluids at week 24 in the combination treated group compared to the control. We also saw anatomical changes at the back of the eye. Here we're seeing an almost 40% further reduction in total lesion area following OPT302 combination treatment compared to the control. And we also saw this reiterated in the choroidal neovascular area, which was also reduced relative to the control following OPT302 combination treatment. These parameters hitting p-values of 0.01 and 0.005. Now, we did conduct a number of pre-specified subgroup analyses. This first one is polypoidal choroidal vasculopathy. We did recruit approximately 18% of patients with PCV, and PCV is estimated to be the most common subtype of wet AMD in Asian populations, and in fact globally, given that the Asian population accounts for about 60% of the world's global population. In patients with PCV present at baseline, we saw a delta or a visual acuity gain of 6.7 letters following OPT302 combination treatment compared to the control. This hit a p-value of 0.0253. And in a pre-specified subgroup analysis by lesion type, we also saw some interesting findings in certain lesion types. We had a very small minority of patients that had predominantly classic lesions. These are the lesions that are typically more responsive to anti-VEGF treatment. This was only 12% of our patients. And for that reason, I'm going to focus on both the minimally classic as well as the occult lesions, which comprised approximately 90% of the patients recruited into this clinical study. You can see very nice separation of our visual acuity curves over time in both of these lesion types. That's shown here, where we saw a 2.7 letter gain in the minimally classic patients compared to the control, and a 6 letter gain following OPT302 combination treatment, which hit a p-value of 0.0008 relative to the control arm. We saw an over 20% increase in 3-line gainers in participants with occult lesions with OPT302 combination therapy, and further 2-line vision gains now in the minimally classic group as well as the occult patient group as well. We saw further reductions in central subfield thickness by lesion type, also in the minimally classic, as well as very profoundly in the occult lesions, as you can see here. And further drying in both minimally classic as well as the occult lesions following treatment with this VEGF-C/D inhibitor relative to the control. This was also reiterated in further drying of intraretinal fluid, as you can see here in minimally classic, as well as the occult lesions. And once again, we saw a very much a reduction in total lesion area, reflecting a change in the anatomy of the lesion, with a reduction in the total lesion area in patients with minimally classic lesions compared to the control when they're treated with the combination, and we saw this also in the occult lesions as well. And again, reductions in C and V area following treatment with the combination OPT302. The C and V area markedly reduced in the minimally classic as well as the occult patient populations. Actually, I need to go back one. What we did show also in another subgroup analysis, I'm not sure I can go back one slide, but then in patients that had RAP absent lesions, we saw a mean gain in visual acuity of 4.4 letters over and above the control group. So RAP lesions that were absent was actually, they did better in this clinical trial. They were more responsive to VEGF-C/D inhibition, as you can see here, hitting a p-value of 0.0025. And then when we look at minimally classic lesions, less RAPs, those where those RAPs are excluded, we see a 4.7 letter delta, a gain in visual acuity of 4.7 letters with a p-value of 0.04 following combination treatment, and a further 6.5 letter gain in those occult patients relative to the control group with a p-value of 0.0005. The safety profile of OPT302 now has been very well characterised. We've really seen no safety issues with the molecule, a safety profile that's very similar to ranibizumab in this particular study, and we now have safety evidence in combination with aflibercept as well. So in conclusion, our phase 2b clinical study met the primary endpoint, demonstrating superiority in visual acuity over ranibizumab with a vision gain of 3.4 letters and a statistically significant outcome of 0.01. This was against one of the most highly performing ranibizumab control arms. Our secondary outcomes were supportive of the primary endpoint, with more patients gaining vision and fewer patients actually losing vision following OPT302 combination therapy. And we saw retinal anatomical improvements, including reductions in CST, subretinal fluid and intraretinal fluid, and greater decreases in total lesion area and CNV area. Following on from having met the primary endpoint, our exploratory and pre-specified subgroup analyses have indicated that our OPT302 has greater activity in those lesion types that are considered to be more difficult to treat with anti-VEGF-A therapy and those with the highest unmet medical need. We've shown promising evidence of activity in both polypoidal lesions as well as minimally classic and occult lesions that are typically less responsive to VEGF-A inhibition. And on top of that, we've shown a very favourable safety profile with OPT302 that is similar to ranibizumab alone. Thank you very much for your attention.